Okay, so today's video is going to kind of preview exactly what we're going to do in Lab 10. And that's essentially just in 3 steps. We're going to isolate DNA from bacteria clumps, those little plasmids, and then we're going to cut those with restriction enzymes, and then we're going to run them on a gel. And the TAs will do that last step for the most part. You'll load the gel, but they'll set up the images and everything. So bacteria cells, remember that they have just like this big, long, circular DNA strand. One other thing that they can collect, even from the outside, is small circles of DNA called plasmids. And these can contain any number of genes. These can be antibiotic resistant. They can cause like different movements in the bacteria. They can be exchanged from the outside of the environment or from other bacteria cells, for example. So this is kind of how bacteria can have like gene transfer and like can kind of breed adaptation, because these can multiply in here and then spread out towards the other neighboring bacteria. But what we do is we actually can control bacteria and what they produce by engineering a plasmid, just a single one, overexpressing it in bacteria, and then using that gene product or that protein, whatever it is, for all kinds of uses. This could make bacteria clean up oil spills. It can produce biomolecules that can cure hemophilia, for example, in people. So this is a great way to mass produce certain things in medicine. So what we'll do first is that you'll essentially be given a tube, and at the bottom there'll be a clump of bacteria, and these will be red right here. So essentially we need to go in here and extract just the plasmid DNA. We want to get rid of everything else. So the first thing that we're going to do is we're going to resuspend everything in that buffer P1. P1 is literally just to resuspend this all so that everything is kind of homogeneous and the bacteria get mixed up. P2 we're going to add, and what this is going to do is this is going to kind of lyse the bacteria. This buffer right here is going to break up the bacteria shell. So essentially at this stage, we have our bacteria cells and we have our plasmids in here. This is essentially going to cause holes to perforate in the bacteria cells so that we can get at that plasmid. So I think this only goes for less than 5 minutes, does this lysis. So you mix with your pipette here, and then here you're just going to invert the tube a little bit. Next you're going to add, this is the last step in this first tube, P3, and this is just going to make everything kind of settle down. This is a neutralization buffer, so this stops the lysis reaction, because this can't go on too long or the lysis buffer will just rip everything apart, including the DNA. So at this stage, you have everything neutralized, just like black like this, and you're going to spin this down really hard for 10 minutes. And what'll happen is that we'll end up with a debris pellet down here from all the bacterial debris that's been broken up. And then we'll also have a supernatant. And so at this stage, we want the supernatant. Leave the pellet at this stage. So don't worry about this. We're going to take this and we're going to put it into what we call the spin column. So this is the column that goes like this, and it has like a little like here, and it's in its own little collection tube. So this is the spin column, and this is where the supernatant from this third step goes in. So essentially you'll let that go in. Oops, I didn't finish that. So don't pour that part out when you get to after step 3. So now that we've thrown this tube away because we took the supernatant out, we're going to spin this tube, spin, and then we're going to add 0.7 mil wash. And essentially what this does is that when you put that supernatant in that spin column, all those plasmids at the microscopic level, they get caught on this spin column. They get stuck to it. It's like a filter, and it keeps them from going down here. Now all the other mess, all the other blue supernatant that we don't need, it's going to go down here when we spin. When we add more wash buffer, let's just call it green in this case, the wash buffer will take out any remaining like bad buffer, just like other stuff that's in there. And eventually we're only going to be left with those little red plasmids, and they're going to be stuck on that spin column filter. It's essentially like a little magnet. So we're at this stage, and we've washed a couple times. We've got tons of plasmid just waiting here. The last step is what we call EB, or elution buffer, because what you'll do is fill the spin column with EB, and what that does is disassociates the plasma cells, or the plasmids, off the filter. And now we would replace that old collection tube with a new tube that's just for plasmid, and we'll do one more spin. And so this is how we took the plasmid from inside the bacteria cell. We broke that up, and then now we kind of hooked it to the spin column, washed it a couple times, and then unhooked it from the spin column, and now we have pure plasmid here. So the next thing that we're going to do is that plasmids are, in a lot of cases now, they're fairly synthetic. We've added certain points in them that we control, certain genes in them. For example, antibiotic resistance in some of these is used as a marker so that they can survive certain antibiotics, whereas the bacterial cells that don't have these plasmids, they won't survive. And that's how we can tell which ones have it. So in this case, we're going to be cutting with two enzymes. Let's see what they are again. AVA1, I don't remember if they're on this side too. AVA2, and then we're also going to be cutting with PVU2. So when I say cutting, this is because there are sites in the DNA here and here for this color, and here, at which when you add the restriction enzyme for AVA2, it goes in here and it cuts that part of the DNA. And now you've cut a part in that circle, it'll cut here too. So now you have this little linear fragment, for example, and if you bring in the PVU2 enzyme, it's going to do the same thing at both sites, and it's going to cut the DNA there. So now we've cut things into 4 different segments by using those two. So this is a common tool because a lot of the times when we cut stuff out of a plasmid, we get rid of a certain thing and we introduce something new, something that's also been cut by those same enzymes, and now it can fuse to those sticky ends if you've seen those. And then this is kind of the beginning of gene insertion, biotechnology, putting genes in certain organisms that can take those plasmids up, and then they'll start expressing that. So for just a little closer look, remember DNA is double-stranded, but there are certain parts of the DNA that restriction enzymes, they cut certain sequences. And this restriction enzymes are actually from bacteria originally, and they cut viruses at their specific spots. So we take these out of bacteria cells now, and they cut at specific spots, and they leave what are called kind of like sticky ends, which means, sorry, this is gonna be kind of hard to draw. If you look up sticky ends in your book, it shows you a lot better how you end up with like 2 ends cut like this. That, like say this goes on. This is just 2 ends of the DNA. And now you can still make that connection there after you separate it with this and then put something new in that's been also cut by this. So get rid of that sticky end, and you bring in another set of DNA that you cut, but this time it's different over here. Now you can seal this with an enzyme called ligase, and then that's just whole DNA now. And so ultimately what you get are plasmids that have been cut here and here, and a gene at another time that was cut here and here, the same little restriction sites, put in, and now you have a plasmid with a gene of interest that you want.